Hello everyone, I am Dr. Akshita and today I am going to start with parasitology. So in parasitology, first we will study about the most commonly heard one, that is ant amoeba histolytica. Okay, so before starting, let's see a few important pointers about it. What does it cause? It causes traveler's diarrhea. Okay. Another important point is it is the second most common protozoan causing death. Third point, the cyst form of this uh, protozoan, the cyst can survive routine chlorination of water. Alright, this can survive it. Next point, what is the habitat? It lives in the large intestine, in the cecum and ascending colon. Okay, also this is a strict human pathogen. It's a strict human pathogen. Now it is present in two forms. One is your cyst form, another one is trophozoite form. Alright, cyst form, important point about this is quadrinucleated. Second is the trophozoite form. Trophozoite form, important point about trophozoite form, this is your trophozoite form. Here you can see this pseudopod. What is the action of pseudopod? This helps in motility of the organism. Second important thing in this is this nucleus. This nucleus is eccentric. That is, it is not in the center. So this is an eccentric nucleus with central karyosome. Alright, this is about trophozoite. Now what the trophozoite does is it enters your through your GI tract. Okay, it spreads by fecal-oral route. So it enters your GI tract, the trophozoite. This attaches to the mucosa and release protease. What will happen? There will be ulcer formation. Now this ulcer is called either your flask shaped ulcer or your undermine ulcer or your water bottle ulcer. You can see the shape here very easily in the diagram, and it's a very important diagram, right? Then what does it cause? It causes something called amoebic dysentery. Now what are the features of this amoebic dysentery? You will have diarrhea along with cramps plus blood and mucus in stool. Alright, another important point is in in males more than females, as well as in people who are rich in cholesterol, who eat cholesterol rich diet, it causes amoebic liver abscess. Okay, amoebic liver abscess, the key point about it is it has an anchovy-sauce appearance of the pus. It's an anchovy-sauce like looking pus in this condition. Alright, this important point. What's the most common site for it? Most common site is your posterior superior surface of right lobe of liver. Now how will you diagnose this? Diagnosis includes one, stool sample. Okay, in stool sample, what you can see trophozoite as well as cyst form. Also you see charcot laden crystals plus RBCs. Now in this diagram as well, you can see some RBCs present in the food vacuole. So these are ingested RBCs by the entamoeba. All right. Next, you can cultivate it. So second is cultivation in Craig's medium and diamond medium sample. Third, ELISA. Fourth, most sensitive one, that is your PCR. PCR is most sensitive test for it. All right. So next is your Naegleria fowleri. Now you can see in the diagram, in the picture as well, it has two flagella. Okay, and it's an amoebic form, so it is amoeboflagellate. This is also a brain-eating amoeba. One important point about it can cause disease in immunocompetent people as well. Okay, immunocompetent people as well. Habitat, it is usually present in soil as well as warm fresh water. Okay, host here, man is usually not the host, man is accidental host. So when people swim in contaminated water, it can go through. How does it go inside? It goes through the nose. All right. So via nose, it goes to the cribriform plate, to the meninges, and then what it causes is called primary amoebic meningoencephalitis. Along with features, which is very common with this, is altered taste and smell. So mortality is very high, as much as 98%. Diagnosis, for diagnosis we have CSF, we see amoeboid trophozoites. Okay, both. Okay, cultivation के लिए we have non-nutrient agar with lawn culture of E. coli, and most sensitive one is your molecular methods. All right. Next is your acanthamoeba. Acanthocytes means spines. Okay, in the diagram as well we can see small spines here. So this is a picture of acanthamoeba. So it again has both cyst and trophozoid form, spine-like. It has spine-like acanthopodia and causes disease mainly of the people who are immunocompromised or disease of immunodeficient. Habitat of it is soil, fresh water, and seawater. All right. Now, how does it enter? It enters through your eyes, nose, and your skin. So through these parts it enters it eyes, nose, and skin, and via hematogenous spread it causes granulomatous amoebic encephalitis. Okay. So it enters through eyes. Also, it causes keratitis, especially in people who wear contact lenses. All right. Then we have Ballantidum coli. So this is a ciliate organism. You can see very small cilia all around it. Okay, this is a ciliate organism. Now, few very important points about it is one, this is the largest protozoan infecting humans. Second, this is the only ciliate infecting human. Third, it is the only protozoan showing genetic exchange in form of conjugation. In form of conjugation. What is conjugation? Conjugation is by formation of a sex pili. Alright. Otherwise, it is usually done by transduction. So habitat, it is in the large intestine. Large intestine. And the infective form is a binucleate cyst. So it's a binucleate cyst, which is the infective form. So what? So once it enters the host, later it will pass through the feces, right? So after passing through the feces, it exits in the cyst form, all right? So it has one macronuclei and one micronuclei. So when the person eat infected food, so it will cause it will go by the oral route and later in the trophozoite form, and the trophozoite form will have cilia all around it, right? So this also what it causes is called ciliate causes ciliate dysentery or balantidiasis. All right, how do you diagnose it? The way of diagnosing is one, again through stool sample. We will see large trophozoites and cyst, both are visible here. Second is molecular methods by PCR. All right, by PCR. Now next is your giardia. Giardia is the most common parasitic infection. Important point. Again, one cyst form and one trophozoite form. Alright, it has a cyst form plus a trophozoite form. Host is human. Important point to remember here that cyst is the infective form. Cyst is the disease form or the infective form. So mode of infection is ingestion of a quadrinucleated cyst. Okay, so now mostly in people this is a self-limiting condition which causes diarrhea, cramps and bleeds and all. Investigation, how do we investigate? Investigation is by PCR. 1, 2, microscopy, so we can see this stool culture, okay, and also enterotest, all of this. All right, so this is your picture of your giardia lamblia. Now we didn't read about the treatment of all these diseases. So treatment, entamoeba and giardia can be treated by metronidazole or tinidazole. Now for entamoeba and giardia, okay, for entamoeba the dose is for 10 days of metronidazole or 5 days of tinidazole. For giardia it's 7 days of metronidazole or 1 day of tinidazole. All right. Also in case of entamoeba, along with metronidazole, there is paramomycin given, which kills its cyst forms. Okay, it kills the cyst forms. Another organism was Fowleri. So for Fowleri, we use, this is a brain-eating bacteria. We use, sorry, not bacteria, brain-eating parasite. So we use amphotericin B plus rifampicin, or we can use azoles, or we can use miltefosine. And for B. coli, we use tetracycline or metronidazole for 10 days. So this was the first part of parasitology. If you like it, then you feel free to subscribe to this channel, and I'd be really happy to make more videos. So thank you.